My name is Pierre, I'm French. You can see this. So I did my undergrad in France in material science and engineering. And then I did a Master of Science at Imperial College in London in neurosciences. And then I ended up here doing my PhD at MIT and working here in Dr. Young's laboratory to treat retinal diseases. The two main retinal diseases we're working on are retinitis pigmentosa and AMD, age-related macular degeneration, because those are the two biggest retinal diseases in the world right now. Retinitis pigmentosa is a disease that impacts a lot of people, young children or old people. You can lose the colors and you can lose the way to see light actually really fast. Macular degeneration is a lot more losing the entire vision. A stem cell is this, just a typical cell that can become anything you want. If you put specific growth factor, specific biology, mechanical stress, anything, you can make any kind of cell you want. Heart, brain, spinal cord, fingers, skin, bone, whatever. So those diseases, when it's happened in the retina, you lose part of the retina, which is called the photoreceptors. And what we are trying to do is to culture stem cells to get new photoreceptors and then inject them into patients. That's the really next stage, but that's the goal of it. When I come to lab, usually around 8:30-ish. First of all is you need to take care of those cells. Add media, change them into a flask from another. So every day you have to do something like this. Usually after that, what we do is make the new experiments we want to do. Can be anything. It can be a flow that we want to run. We want maybe to sort cells, to do an in vitro study with gelatin or with the biomaterials. We can do an in vivo study because it's the time to inject on rats. We have to do all of this before even thinking about doing human studies because it's not FDA approved. Everything has to be done in the right way in order to reach a treatment at the end of it. Cruelty is horrible in animals. We need to take care of everything we're doing. Everything is really important. It has to have like rules all the time. I totally agree with this. It's just the 1% is just that for researcher, for us, it's really hard. Every in vivo studies has to be like it's really long. We have to take a lot of precaution. If you can get the right stem cell, the right biomaterial, and the right growth factor, you could possibly cure anything. It will take time, obviously, because there is a lot more than just doing research in a lab when you want to have a treatment at the end. But I really hope that what we are doing here will go to maybe a clinical trial one day or to a future treatment for blind people. Yes, I really hope so. That's my main goal when I wake up every morning and come to lab, actually.